Here is a step-by-step procedure to resolve a dihybrid cross problem. In fact, it is problem number 5 in the practice of Bio 196, students of Bishop's University. So the first step is to, of course, identify the problem and make sure that you have really a dihybrid cross in front of you. So this is really easy. You determine if it is a monohybrid or a dihybrid cross by looking at the number of traits that are followed. So if there is just one trait with two versions, like for example the color of a fur of an animal, and if the color is spotted versus non-spotted, for example, or if the color is black versus white, it is a monohybrid cross because you just have one trait with two versions. If in contrast you look at two traits, so for example in flies you will look at the body color and the length of the wings, and each of the trait has two versions, then it is a dihybrid cross. So here we will cover the dihybrid cross. If you're not sure how to solve a monohybrid cross, I suggest you read the first video that I published already. So here, this one is really about the dihybrid cross. So the second step in the problem is to identify the dominant and the recessive alleles of the two genes, and after that to determine the genotypes and phenotypes that are related to these genotypes. So at this moment, you should read the problem and underline the genes and their alleles. Then you should try to determine which ones of the alleles are dominant and which ones are recessive. So if you read the problem here, we're talking about two different genes. The first one is the ability to taste a drug, and this is carried by a dominant gene. And the capacity to, in fact, the non-capacity to taste it is recessive. While you are looking at this particular trait, so the capacity to taste the drug, you are also looking at the pigmentation of the skin, and you learn here that albinism, which is an absence of pigmentation, is carried by a recessive gene. So if you look at the two genes and their versions, you have first the gene 1 that is the taste, and you will use the letter T for taste. The capacity to taste the drug is dominant, so we will identify it with a capital T. The taste blindness, however, is recessive, so we will identify it with a small t. Then you will follow also the transmission of a different gene, which is the albino gene. So it is a complete absence of pigmentation. Because it is a particular trait called albinos, we will use the letter A, and the normal pigmentation is dominant, so we will use the big A, while albinos is recessive, and we will use the small a. So when the genotype of a certain individual is big T or big T small t, it gives you a taster phenotype, so a phenotype that can be called big T. And a small t genotype will in contrast give you a non-taster phenotype, so a person who is unable to taste the drug. If you look at the pigmentation of the skin, the genotype little a gives you an albino phenotype, or small a, and the genotype big A or big A small a will give you a normal pigmentation identified by a big A. Now what you will want to do, you will want to identify the individuals that are presented in the text. So you will simply reread the problem and underline all the individuals that are mentioned and identify their phenotype, establish, if possible, their genotype. So the first individual that we are talking about here in the text is the woman. It is a woman who is normally pigmented and taste blind. So the phenotype of this woman is big A small t. So big A for normally pigmented, small t for taste blind. Her genotype is not fully determined because the big A phenotype can come from big A small a or big A genotype. So as a result, she can be either big A small t or big A small t small t. The second individual that is mentioned in the text is the father of this woman. He is an albino taster, so his phenotype is small a big T. His possible genotypes are small a big T, or small a big T small t. If you don't understand why we're changing the t's here, you just have to refer to the previous slide that showed you that a dominant phenotype for the T will result of a double big T or a single big T genotype. Then the third individual is the man that she will marry and with whom she wants to have kids. So the phenotype of this man is an albino taster. So he is small a big T. So the possible genotypes are exactly like the father, small a big T, or small a big T small t. And the fourth individual that is mentioned is the mother of this man. This woman is known to be taste blind. We don't have any information about her skin pigmentation. So what we can say for her, we can say that her phenotype is small t and her genotype is small t. Now the next point will be to identify who of these individuals will want to mate and have kids. So we know that this is the first individual, so the first woman who will have children with the man, and we want to know the genotypes and phenotypes of the children that they possibly can have. It doesn't mean that they will have all these possibilities, but they can possibly have different types of children, and that's what we want to determine. So what do we need to know now? We need to establish more precisely the genotype of these individuals, so the woman and the man identified in this problem. So first of all, the woman, she has an ambiguous genotype because we learned that she was big A small t, or big A small t. So we have to determine which one of these two possibilities is the real genotype of this woman. So we don't know the genotype of her mom, and therefore the gametes from the mom are unknown. But we do know the genotype and the phenotype of her dad. We know that the dad is small a. We know that the dad has gametes that, or produced gametes that can contain only the small a gene, because the dad doesn't have the big A, so he will contribute only gametes with a small a. Therefore, this woman has forcefully at least one small a coming from the dad, and the second A is a big A because she's not albino. So forcefully, she is big A small a genotype because she has a normal pigmentation, but she forcefully received a small a from her dad. Regarding the man is either small a big T or small a big T small t. Same thing here, we learn from the text that the mom is taste blind. We have no information about the skin color, but we know that the man anyway is small a in any circumstances. So the only thing that is ambiguous for him is the taste capacity or the taste blindness. Because the mom was taste blind and this is a recessive gene, then we know that her gametes contained, all of them contained only small t. So she had no gametes that could contribute a big T to this man. We have no information about the dad, and the gametes of the dad are unknown, but we know that the man is carrying at least one big T because he is taster. Therefore, we know that the man has forcefully a genotype that is big T small t because he received an unknown gamete from the dad, but in fact this gamete must have carried a big T because the man is taster, and the mom could only contribute small t or non-taster gametes. So now that we have the precise genotype of the two parents, we will be able to determine so we determine their genotype, so we will need to determine their gametes and the chains of production of these gametes. So here, if we look first at the woman, we know that all the gametes will contain a small t because it is the only option for the t. So the t here is not really important, it's just the a that will be different in the gametes of the woman. And because we know that the two genes are sorted independently, we know that the small t will have an equal chance to be put with a big A or a small a. So on average, you will have 50% of the gametes at meiosis that will contain big A, small t, and 50% of the gametes that will contain small a, small t. It is a bit the same thing for the father or the man. So we know that all the gametes will contain a small a because that's the only option he has. But for the T, we'll have equal chances to put a big T or a small t with the little a. So at meiosis, the gametes that will be produced will be 50% small a big T, 50% small a small t. So from this, we we can now sort the problem and determine the genotypes, the possible genotypes and phenotypes of the kids. Here, a little precision in this text was that we were not allowed to use the Punnett square. In the practice, I didn't mention that, so you can also use the Punnett square if you feel comfortable with it. I will cover here the fourth-line method. So this is the way we do when you don't use the Punnett square. So again, the woman has big A, small t as a genotype, so she produced 50% of gametes containing big A, small t, and 50% of gametes containing small a, small t. She can mate with gametes of the man that have 50% of chances of being small a, big T, and 50% of chances of putting her gametes with a small a, small t gamete from the dad. In the case where you have the gamete big A, small t, so half of the cases, put together with the small a, big T, then what will result is a genotype for the kid of big A, small t, big T. This has 25% of chances to occur because you basically have 50% of the 50% possibilities of having this genotype. Same thing, so it will give us a phenotype of a normal taster kid. So 50% of the offspring of this couple, on average, 25% of them will be normal tasters. Now, the second possibility for the gametes big A, small t of the woman were to be put together with a small a, small t gamete from the dad. So in this case, you have a genotype that is big A, small t, and this is again a 25% of chances to occur because simply it is 50% of 50% of the cases. Like 50% of the gametes of the man will on average have 50% of chances to find this gamete of the woman. So again, 25% of the kids on average can be normal and taste blind, normal skin color and taste blind. Now, the second half of the woman's gamete are small a, small t, and they have again 50% of chances to be put with a small a, big T gamete. So in this case, the kids will be small a, small t, big T, so albino tasters. This occurs in 25% of the cases. And the second possibility is that this little a, little t gamete of the mom is put together with another little a, little t gamete from the dad, so you will result in a genotype that is small a, small t. So again, you have 55% of chances that this condition occurs and produces albino taste blind kids. Now the last thing you have to do is to double check. So what were you asked to do? You were asked to identify the possible offspring. So this is the qualitative results. You should give the phenotype and or the genotype of the expected offspring. This is what you did because you gave the genotypes and the phenotypes. Calculate the ratio of the types of offspring. You were not asked to do that, so you don't have to do the ratio. However, you were asked to calculate the probability of obtaining one specific or all of the specific offspring. So here you have given a percentage. So this is what you had to do. You gave quantitative results as well as qualitative results. Are your calculations correct? If you add all the probabilities that you have calculated, you should add up to 1. And here we had 4 times 25%, so it adds up to 100%. So this is correct. Our calculations are probably okay. This completes the solving of this problem. Thank you.